And it's my pleasure to have Ivan back from the UGENE project. Those of you who have been regular BOSC attendees will have heard about this before, so we'll find out what they've been doing this year. Thank you.
Hello everyone, my name is Protsyuk, and today I'm glad to represent the team of the Unipro UGENE project that is developed by the Unipro company in Akademgorodok of Novosibirsk, Russia. So the theme of my presentation is shared bioinformatics database within Unipro UGENE. Here is the plan of my talk. I'm going to start with a brief description of the UGENE platform, then I'll go into details of our recent shared database related functionality, tell about its general concept, as well as some technical questions and quantitative characteristics. And finally, I'll share with you our future plans and prospects. So Unipro UGENE is a desktop open source bioinformatics toolkit. It is being developed in C++ language using the Qt framework, so it is cross-platform as well. UGENE comprises a number of popular bioinformatics algorithms, supports more than 20 widespread file formats, and offers interactive graphical views for different objects such as annotated sequences, multiple alignments, genome assemblies, and so on. Moreover, UGENE provides its own workflow designer for arranging required tools and algorithms into pipelines. The designer, by the way, can be easily integrated with Galaxy. And it also stands to mention that UGENE has the NGS framework that was partially developed in collaboration with National Institute of Allergy and Infectious Diseases. And now let's get closer to the topic of my presentation. The efficient storage of biological data is an ever-present issue. It is not only about large data sizes, but also about the requirement of simultaneous access to them by several scientists. For example, a few researchers in a lab may need to work on the same data, like a set of primers or data produced by sequencing. And in this case, the information has to be updated and synchronized between different users and kept in a common storage. Some notable implementations of such systems already exist. For example, CLC Bioinformatics database or Gene's shared database, but they are proprietary and quite expensive. Thus, in the latest UGENE version, we have introduced a new functionality, shared bioinformatics storage. So far, it is represented by an ordinary database. Currently, we support MySQL. To start sharing data via UGENE, users only need to deploy a public database server. After that, any UGENE user can connect to it and access stored information, add new objects, or delete existing ones. In fact, from the user's point of view, the storage looks like a usual file opened by UGENE. The entire content of the database can be viewed exactly the same way as if the data were located on a local computer. Objects in the storage can be organized using folders, as well as they can be moved between different folders, other connected shared databases, or local documents using conventional drag and drop. And UGENE instance, in turn, constantly monitors the state of the database and shows changes made by other users. And now let's consider a few technical questions. So what does really distinguish our system from its counterparts? It is definitely our approach to data storing. Normally, entire serialized objects are stored either within tables of a database, as it is implemented in the Gene's package, or directly in the file system of a public server, as in CLC Bioinformatics database. In these both cases, if an object is accessed from a storage, it has to be downloaded completely to the client side. And instead of this, we have developed our own fully relational data model for storing all data types supported by the UGENE platform. Therefore, the data can be downloaded to the client side in pieces when they are really needed. We divided available biological types into two large groups. The first one, in the right column, includes complex objects with that may have a size that reach a few gigabytes, and it is undesirable for such data to be transferred entirely via the network. So we have developed individual data models for each of these data types according to their internal structure. The main sense of these models is that they allow achieving a deep data separation so that that objects can be downloaded to the client side in pieces. And in this way, the system can significantly decrease both network traffic and the client application response times in comparison to analogous solutions. However, such complex individual approach is not always necessary, since some data types in bioinformatics deal with objects no more than a couple of megabytes in size, and they actually constitute our second group. So these data are allowed to be passed in their entirety via the network, and for such objects, a generalized storing model is used. Unlike the previous one, this approach implies a bulk storage of serialized data, but since they are small enough, this technique is fully justified. Now it's time to talk about some quantitative characteristics. The shared storage is capable of storing millions of biological objects. Entire public databases such as GeneBank or Protein Data Bank can be easily imported into it. Large arrays of next-generation sequencing data are supported as well, such as genome assemblies, and can be browsed remotely. And as for single objects, for example, it takes around a minute to upload a sequence with length of 100 million bases to the shared storage. You can notice that this is quite longer via the network. But such data, once uploaded to the storage, can be accessed almost instantly by every UGENE user subsequently. And the described shared storage allowed us to implement another feature in UGENE, a public shared storage. Currently, it contains commonly used genomes and plasmid sequences accessible from within UGENE. And internally, the storage is a remote database with a read-only access for everyone. So every UGENE user can connect to it and use the data that we supply. And in conclusion, I would like to share with you our future plans and emerging opportunities. Our short-term goal is the optimization of the system. This includes performance improvements for some cases, such as a large number of shared objects. And also, I want to mention one of our long-term goals that is now closer. The goal is to develop a distributed execution system. You may wonder how it is actually connected to what I told about previously. Well, currently our workflow designer can operate only locally. But since we have a shared database from now on, it can be used for data exchange and synchronization between separate workflow blocks that work on different machines. Another possibility is to apply data parallelism during workflow execution. Besides, I want to say that my team and I are looking forward for the feedback on our shared database from the community, and the most popular requests will be included into the next UGENE major release. Also, we are open for collaboration and ready to help everyone install and tune the shared database for their needs. Finally, I would like to provide you with some useful links. Thank you very much for your attention, and if you have any questions, you are welcome.